Characterisation of whisker use in dormouse locomotion, climbing and gap crossing. The hazel dormouse is a shy, mostly arboreal and nocturnal rodent from the Gliridae family. A recent report by the People's Trust for Endangered Species estimates that dormice have declined by 38% in the UK since 2000. This huge loss demonstrates how important future conservation management is. To save the species, we'll need a good knowledge of their habitat requirements, such as how they move around and use the tree canopy. Being active in the dark and navigating precarious treetop routes means vibrissae, or whisker use, is particularly important in dormice, and a previous study has found touch to be their most developed sense. However, comparatively little is known about the physiology and sensory ecology of dormice, due to their shy nature and protected status in the UK. Limited studies in Europe have addressed landscape-scale gap crossing, but not small-scale locomotion. I sought to characterise whisker use with a similar methodology to other active touch sensing research. Filming in a top-down perspective is a standard technique for whisker studies on mice and rats, allowing direct comparison to my data. To understand how dormice use their whiskers during normal locomotion, and how they might use them while navigating through the tree canopy, I also filmed side-on footage. My third objective was to quantify the dormouse's ability to cross small-scale gaps as a proxy for jumping between branches. A high-speed camera was used to film top-down footage of locomotion in an open arena lit by infrared light box. Climbing substrate, such as twigs, was added and the same camera used to collect side view footage. I designed and adapted the experimental enclosures, ensuring the feeding platforms were too high for the dormice to reach them directly. A retractable pole between the raised platforms created a gap, which was increased each night, with food used as an incentive to jump. An infrared motion sensor trail camera was set up above the enclosures to film jump attempts overnight. I used MATLAB Biotact whisker tracking tool on the top-down data, which revealed figures of 21.8 Hz for frequency, 124.27 degrees for the mean angle, and 29.7 degrees for the amplitude. Side view, amplitude, frequency, head orientation, whisker position, and whisker spread were calculated with a manual tracker program. Statistical analysis indicates that dormice do adjust their whisking strategy for different types of locomotion. However, there is significant overlap and similarities, meaning it is difficult to define distinctive jumping, walking, or climbing whisker use profiles. I did find a reduced amplitude and a higher head orientation in jumping, and that the position of whiskers was different during climbing compared to walking or jumping. Frequency and spread were similar between all conditions. The dormice were able to jump at least 21 centimeters horizontally. Calculating their forward reach revealed that both dormice repeatedly jumped gaps too large for them to palpate the opposite side with their whiskers. This differs from previous research on rats, which found them unwilling to jump across gaps larger than their whisker reach in the dark. Many mammals with vibrissae use them only for passive data collection. My research shows that, like mice and rats, dormice actively control and adjust their whisker use depending on task. Whisking frequency is also similar to mice. I found that dormice use a look-ahead strategy when walking and jumping, where whiskers are held further forward and whisking amplitude is reduced. This serves as an early warning system when information about obstacles directly in front becomes more important than general data from sweeping the whiskers back and forth. During climbing, the dormice angled their whiskers down towards the substrate. This additional contact with the surface could provide vital information for stabilization, balance, and guiding foot placement. The dormice demonstrated an impressive jumping ability, but also hesitation or refusal to cross some large gaps. Such behaviour would impact on predator avoidance and foraging efficiency in the canopy, indicating how important dense habitat connectivity is. Footage shows the dormouse successfully jumped the gap on the first try, even once it was too large to palpate, but the mechanism for this is as yet unknown. My experiment took place in a windowless room lit only by the trail camera's infrared light. If the dormice were able to see across the gap, it suggests their dim light vision is much better than previously reported. My MSc project undoubtedly created more questions than answers, but it did provide some interesting preliminary insights, which inspired a paper by my supervisor, Dr. Robin Grant, which was recently published in the Journal of Comparative Physiology.